Well, hello everyone. Good morning, good afternoon. Welcome in from the continent and beyond for another very exciting Exploring by the Seat of Your Pants broadcast. Now, if you're new to us, we are all about bringing conservation, adventure, and science into classrooms around the world, and everything we do goes to our YouTube channel. So if you want to check this one out in 3 weeks or 3 years, or check out like 4,000 other broadcasts, there's lots of opportunity for you to explore and discover all sorts of cool things. So, first and foremost, thank you so much for tuning into this as part of our Future with AI series. We did another broadcast just last month. You can check that out on our YouTube channel. And we've got another one smack dab near the beginning of January, so you can follow along with the adventure. Now, this series is a collaboration between us at Exploring by the Seat of Your Pants, the amazing folks producing it at E2 Adventures, and powered by Digital Moment. We've also done a bunch of other programs with Digital Moment that, again, all of which are on our YouTube channel. So today we've got a whole horde of amazing speakers from all sorts of industries to highlight with you some really cool videos as well. I'm very excited about it, and I hope you are as well. To begin, I'm going to bring in Sabrina from Digital Moment to tell us a little bit about all the amazing stuff that they do. So I'll zip it. Sabrina, welcome to the broadcast, and tell us some more.
Hi everyone, I'm Sabrina and I work at Digital Moment, a Canadian nonprofit dedicated to supporting educators and youth with the digital skills and critical thinking they need to thrive in a rapidly evolving world. During today's field trip, you will probably hear terms like computer vision, data, models, and digital brain. These ideas are fascinating, but can also be difficult to understand. That's where Digital Moment comes in. We support educators with hands-on learning experiences that break down these concepts in meaningful, classroom-ready ways with free workshops and lesson plans. To learn more, visit digitalmoment.org and sign up for our newsletter to stay updated on all our free programs and upcoming free sessions. Enjoy the rest of the field trip.
Thank you so much, Sabrina. And now we're ready to dive into today's exploration of the systems, the people, the machines that make sure you function at your best. So every day we go about our normal lives. We go to school, we go to work, we come home, we visit family and friends. But if you've ever gotten sick, and who hasn't, I had a cold just a couple weeks ago, then you know that all these normal activities can come to a grinding halt when we're not feeling our best. So what happens next, and how do we get back on track? The world of healthcare is huge. When you're sick, you see a doctor or nurse, you go to a clinic or a hospital, you might even need surgery. I had surgery just a few months ago, unfortunately, for the first time. You might get medication from a pharmacist, like these medications I have here, ibuprofen, a little Alka-Seltzer. In Canada alone, there are over 7,000 healthcare facilities. Just look at this map. The red crosses are hospitals, the yellow crosses are clinics, the purple crosses are nursing homes and long-term care facilities. And within each of these healthcare facilities, there are doctors and nurses who have gone through years of training, machines and medications that have been tested and retested, all to diagnose and treat you when you're sick. Today is all about how AI and technology are changing the way people, machines, and medications come together to keep you safe and healthy. So over the next 3/4 of an hour, we're going to take a look at 3 parts of this big system that is healthcare. First, we'll visit an AI startup called Scopia, where they're inventing a tool giving surgeons superhuman vision in the operating room. Then, we'll visit a lab that makes sure the medicine you buy in the pharmacy is safe and works as promised. And lastly, we'll visit an innovator who's using AI to come up with new medications faster. Listen closely, because at the end of today's event, we'll have a friendly competition with a Kahoot quiz. I'll pull up this game pin several other times and I'll share it in the chat, but if you want to pull it up in a separate tab now, we'll get to that at the end of the broadcast. All right, we're going to go to Montreal, Quebec for our first stop at Scopia. Gabriel, welcome in. I've heard you've created an AI-powered tool to help surgeons in hospitals. Can you tell us more about it?
Yeah, absolutely. So we are a small startup that is using artificial intelligence as well as something weird called computer vision to help surgeons have a superhuman vision. So be able to help them see things that they cannot see to the naked eye. And that whole experience starts with something fancy called an MRI. So if you can change the image and show us what an MRI looks like, there you go. So hopefully nobody on this call will know what that is, but if you ever need surgery one day or you are sick, we are using special machine called MRI or CT scans that are able to see through our bodies. So we're able to see the interior of our bodies, the anatomy, and If we have cancer, tumors, or special sickness, we can see through our bodies. However, the result of that scan is quite complicated to understand. So if you move on to the next image, please. Surgeons are looking at this kind of black and white image that is showing them the interior of our body, and it's quite hard to understand, as you can imagine. All of our bodies, while we may look, we're all flesh and bone, we're all made very differently on our insides. We come in different shape and size, and our anatomy is not always in the same way. And so surgeons are using these complex imaging to try to better understand how the human body works. But it's still quite hard for them to understand. And so Scopia is developing a special tool that is able to give them a better understanding of the anatomy before they go inside the operating room, but also that gives them real-time assistance. So what we do is that using artificial intelligence, we're able to automatically understand all the anatomy that we have on these pre-operative scan, which is what you see on the left. So all the color codes are different pieces of anatomy that we can automatically label and see with AI. And then we're going to do a very high resolution 3D model of that CT or that MRI, which is what you're seeing on the right-hand side here. So now the surgeons can really understand where are positioned the arteries, the veins, the bones, our bladder, which holds our pee, all of these things. So it really helps them better understand the human body that they will soon operate or work on inside the operating room. And perhaps we can move on to the next image. And what we like to do with that is that now it may seem like it's something that's completely inside the future, but we have across the world, there's about 10,000 active robots in the operating room that are performing surgery every day. And so these robots, as you can see, are very high technology pieces of equipment that they're not performing robotic in a autonomous fashion. They are being controlled by a surgeon, but imagine that you're playing a really cool, complex video game where using a small controller, you're able to control a surgical robot to perform actual surgery. Now, these robots are really impressive, but they cannot operate on their own. And using them by the surgeon is quite hard. So we are developing a GPS, if we could say, to better help guide the surgeon in real time while they're operating on the robot. Back to you, Jesse.
Thank you very, very much, Gabriel. What a cool looking thing that is. My goodness. All right. Well, thank you so much for that fascinating look at AI in the operating room. It's crazy to think that one day there might not even be a human doctor in the operating room, an AI-powered robot that could perform the surgery, stitch us up, and send us home with a prescription. So what kind of prescription might we need after surgery like that? Maybe something like the drugs I showed earlier. I don't know if you've ever had to use these at home, but certainly helpful to have some things at home in case you have stomach, headache, all that unfortunate stuff. All right. Did you know that all over the world every year people take about 50 to 120 billion pills of aspirin? How do we know that medicine like this is safe to take? How do we know that it'll work? Today, we are visiting Neopharm Labs in Blainville, Quebec. Companies like theirs test new medicines again and again before they can be sold in your local pharmacies like Jean Coutu or Shoppers Drug Mart. Today, we're going behind the scenes to see just how that is done. So a big welcome in to our friends at Neopharm Labs. Hey guys. Come on in. Unmute your mic. Someone has to be muted every call. Otherwise, it's no fun. No worries. There we are. We're set.
There we are. All right.
Hello.
Hello, everyone. So my name is Jean-François. I've been working at the Neopharm for quite a couple of years. Neopharm is a company that is specialized in testing all sort of pharmaceutical products. They may be pills, they may be syrups, they may be injection liquids that are injected by your doctors. So here in Neopharm, we have different labs, all equipped with high-tech instruments that allows us to do all these analysis. So I'm going to run you through the first lab here that we have. It's a lab that we call a chromatography lab, where the specialty is to separate your components, your active ingredients, in other words, the drug that you're taking, like we said earlier, aspirin or maybe Tylenol, and we need to extract the drug from the pill and analyze it in one of the systems that we're going to show you to make sure that we have the right amount and that we have the right drug, because we don't want drugs to be mixed up. So the first system we're going to show you is this one here in the end. It's called an HPLC system. We can see it right now on the movement. So it has a robotic auto sampler that moves around all these little tiny vials like you just saw, and it's gonna grab it. There's liquid in these little vials that we've prepared and put in the vial that contains the sample, the part of the drug. So we take the pill, we grind it, we extract it with a solvent, and we put it in the vials here. And then the auto sampler comes and pick up the vial. And there's another here. This is an injection port. So there's a needle that's going to grab the liquid inside the vial and inject it right here. We don't see it, but it's in there. And the liquid will go all the way down here in what we call a column. So it's a little piece. I don't know if you can see it. Maybe we're to try to bring it up. So you see there's a column here. So basically that's like a tube that is filled with particles that will separate each component that is inside. So if like when you take Tylenol, there may be some other ingredients in there, maybe not Tylenol, but I know that some Advil sometimes have caffeine in there because it helps the body absorb the drug at a faster rate. So what this will do is they will separate both ingredients, and we will be able to detect them in the detector, which is one of the two that are here. And then we use a computer with the software to the analysis and the calculate the exact amount of sample drug that we have in each sample that we test. So this is a very widely used instrument for many purposes. It serves other kind of analysis, but its main use is this. So we'll be moving on to another test a little more. So this is a lot of automated technology, but we do have a lot of testing that needs more handwork. So we have technicians that need to do some extractions to verify some impurities in the sample. And if we're lucky enough, we should see an analyst doing a live extraction with a sample called a xanthan gum, which is used as a thickening agent, either in food or even in the pharmaceutical industry. And this test has different solutions of color, and the extraction is to detect a contamination of lead. We're going to check if the analyst has I think we're good.
Ooh.
We're good.
We can go back. We can continue.
Yeah, there's a lot of going on in the lab today. Well, every day is a very busy day, so hopefully we'll speed up a bit. So
on that note, actually, how many people do work at the lab?
So inside the lab, I think we have over 60 people. It's maybe 60 or 80 analysts. We have in a research and development team, and we have a microbiology lab, all with the chemistry. It's maybe about 80 employees, maybe a bit more.
As you continue, I've just got to say, what I love about this is that this is when people envision science in their mind. It's exactly what you're walking us through, which is so unusual for us at Exploring by the Seat of Your Pants. But to see it like 50, 60 people in lab coats actually dealing with things in beakers is quite fascinating. Thank you so much for this.
That it needs a lot of space. So he's preparing. We're not actually doing have ready for the setup, but we have all the solutions. Maybe you don't see it, but we have different kinds of solutions of different colors, and he will put the sample in there and do extractions. And as the extraction it goes on, the color will change to so we can detect the presence of lead, which is a contaminant that we don't want in the sample. So this is a test going. Unfortunately, maybe the timing is a little bit off, but we have something else interesting what we'll show you. We maybe come back later if we have more time. So the next lab we're going to show you, it's a lab where we test the dissolution of any drug that you need to ingest. So we'll have a little bit of playing around to do. So whenever you take a pill, you need to make sure that when the pill is injected, that it's fully dissolved in the stomach as it's intended. So you don't want to have a pill that just goes in your stomach and stays there. So we need to do an analysis that we call a dissolution, where you put the pill in an acid medium, and then we'll mimic the stomach digestion.
Yeah.
And this is where we'll be able to determine if after a certain amount of time you receive the right dosage. So these are the bath. We have different setup. There's one that we're lucky but not lucky at the same time. There's one running at the current moment, but it needs to be protected from light. So it's all covered, so we cannot see the actual content inside. But my colleague here, Christina, will do a little demonstration. I cannot do it myself because I don't have the access to the instrument, because I haven't received the training, all of which is very important. So she's logging in right now with her user password protected.
Yeah, don't look, shield your eyes.
Look, so you won't be able to see anything. And there you go. So now she got access, and she's simulating. That's not how it works usually, but you see that there's paddles that are rotated. So normally these will be down below here. And what happened is that each of these, there's 6 vessels. The vessels are filled with a liquid. Usually it's an acidic liquid because we want to mimic the stomach. And then what happens is we drop one pill in each of the vessel. The paddle starts turning. And then after a certain amount of time, we will withdraw the liquid and analyze in one of the big machines that you saw earlier. And we'll be able to determine if we did get the drug in the right amount of time, in the right amount required to do the
I've got to say, I've literally never thought of this, that you've got to make sure the drug gets to where it needs to go and is dissolving at just the right spot. Like, I've never even conceived of that. So
yes.
So my question that might preempt a kid question, and I'm sorry for that, classrooms, but why? I mean, the stomach, of course, your actual stomach inside your body doesn't have light hitting it, but how did we even figure out that you couldn't have light hit it? Like, is it going to break down the pill in a different way than it would if you had light on it? Like, it's so interesting.
Well, what do you mean?
Well, so you said when it's in the actual chambers, you can't see it because we've made it like where it's I guess not a transparent plastic bag or something where you can see what's actually happening to the pill. Do you do that because you don't want light to be hitting the pill as it's going through the tract? Or
for the one in the, yes. So this is the molecule that we're testing here is light sensitive, so it's gonna degrade if it gets in contact with the light. So that's why we need to cover it. But most drugs aren't sensitive to light. So this is a very special case. But normally we can see what's going on in there, and you would see the pill that drops at the bottom, and you start to see a small vortex of particle that starts flowing around and dissolving around, and then it gets fully dissolved, and then you use that solution to test in the HPLC machines that we showed you earlier.
Super cool. Thanks, Jean-Francois.
Yeah. So, and this, there's a second phase to that. So we do the stomach phase, but some medicals need to be taken on an extended release. So if you want to have the action too long for more than 1 hour, then you have a second stage where you change the solution and you do even more testing. This is a bit more complicated, but it's just to remind us that Some meds are intended for immediate release, like when you do take an Advil or an acetaminophen, you
Yeah.
want it to be active right away because you're in pain.
Yep.
But when you take some other medicines, you want them to last the whole day. So this is where you have a controlled release of drug, and it's a complete. We use the same machine, but there's additional steps that are involved that we need to test to make sure that it will last at least 6 hours as intended, for example.
So, I mean, if we get this wrong, of course, the drug's not working as it's intended. It doesn't help your headache. It doesn't help your stomach ache, what have you. But how do we make sure that like a billion pills all have the same coding? Like if we find out that, oh, it takes 1 hour to digest, how do we possibly like control that for such a huge volume of drugs? It's so interesting.
Well, this is it's all now we're getting a little technical, but there's a statistical analysis that needs to be done to represent the batch that is made. When they prepare millions of pill, maybe making a batch can take 1 or 2 days to make these million pills. But every time you make a batch, every time you need to make the dissolution test. And this is one of the tests, but there's a lot of different tests where you do even more testing on more than, so here we tested 6 pills, but there are tests that we're going to test 10 pills, 20 pills, 30. It depends on what's the goal of the test. So once here it's statistically accepted that we demonstrate on 6 pills that it will represent a batch, but there are other tests that you need to do on more than 6 samples. So yes, the more the more that the pharmaceutical companies make, the more tests that they need to do.
Yeah, fantastic. Well, thank you so much for that. I know that's a complicated thing to explain, but really appreciate it.
So we will go back to the chemistry lab. I'm not sure that we will have the extraction test. I think it's maybe a little technical. If not, we have something really cool to show you in the back of the company. There's an RD lab where we have very high-tech technology. We have take a little bit of a walk here, so we may fasten the pace a bit. But so this R&D testing lab has a very special room in there where we have the most high-tech technology here on site, and the most expensive, obviously. When you talk high-tech, you talk expensive instruments. And of course, these instruments are they look kind of similar to the first one I showed you, but there's another part in the instrument that's completely new. And this other part, it's a very special detector that allows us to measure components at very small concentration that you cannot see with the eyes or any other kind of a technology. So, and the goal to use these instruments is to search for contaminants. One contaminant that I can talk about, and I have an example here. Well, it's not a real example, but a water bottle. So whenever you use plastic as a packaging component, it could be for your pills, it could be for syrup. Obviously, in the food industry, plastic is also used. The companies, even the food industry, have to make sure that the plastic they use are safe, because we don't want to have microparticles of plastic that gets dissolved in either the food or your meds, because if that happens, then it's not good news for your body. You cannot process well plastic, and it can lead to very side effects or other health issues. So you're taking meds to help with specific health issues. We don't want these meds to cause other issues. So that's why testing for micro plastic is very important. So, and this is, I should say, relatively new to industry. Maybe it was introduced 20 or 30 years ago. Before that, it was never tested because there were cases, there were studies that demonstrated the presence of plastic in water bottles. I think that was the first case where it actually, I think it killed very a lot of people. So we had to make sure that plastic was safe. So we're getting there. So this is the R&D lab, and the little room here that you see, it's a very noisy room, so you might hear a bit of a background noise, but we should be fine. So we have 4 instruments here. 3 are identical, and one that's a little bit different is the one here in the back. There's the difference is with the big column here. It plays a very important role, and this is the main one that we use to detect the micro components plastic. So here you see on the screen, on the video, a simulation of the sample. So once we do, when we extract whatever component that we don't want to be in the sample, we introduce it in the system. And all these little particles, you have to think very, 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 very small. These are molecules in very, very small amount. Like I said earlier, you could not detect those in EDI with any other known technology. This is the technology that is used for that because it has a higher sensitivity. It can detect a very small concentration of almost anything that we want. And here it's used mainly to detect the presence of plastic components that could be in the meds. The studies that we're conducting here is not on the meds themselves, but before that, because of course, we don't want the meds to the drugs that you buy to have been in contact with plastic that has low quality. So what the companies do is they select the packaging, the bottle that I'm going to show you anything. Let's say I want to buy this to put my solution in there. So they will send us clean empty bottles, and we will conduct a study. We will use different solution to stress the bottle to get as much plastic as we can out of there, and then we will analyze it in this instrument, and we will be able to say, is there something? If there's something, what is it? Is it harmful, or is it at a concentration so low that it's actually safe for the human body?
I'm really happy. Like, it's really neat to know that you guys do all this, because I know when we do our ocean plastics programs, like there's a lot of plastic that gets into the ocean. There's a lot of plastic that gets into fish.
Sadly, yes.
Concern of classes and a lot of the shows that we do. So to see that there's this industrial scale effort with like extremely expensive machines designed to make sure that we do not introduce contaminants to people is really impressive. So thank you for all this.
Yes.
I know you might have some more to share too, but we do have a bunch of questions coming in, which is exciting. So from our last section, one of the classes, Mr. Faulkner's group at our 2nd Street grade 4-5s, they want to know what does happen if you have a pill that doesn't dissolve fully in the stomach. Is there any harm that comes from that, or is it just not being effective?
So I'm gonna talk more test wise because I'm not a doctor. So what's the impact on the body? I don't want to say something that's not true, but so you have more than one stage. If there's an issue when you do you test the first 6 samples, you move to a second stage where you add another 6 samples, and then you can move to another stage again, and you add 12 more samples. So and that when that happens, then you have a more representative batch, because you may have one pill that had a little lower disintegration rate. Let's say it should be normally dissolved after 30 minutes, but maybe this one, for whatever reason, it would have dissolved after 35 minutes. So on a human scale, 5 more minutes is not going to be necessarily a big deal for the patient, but for the pharmacy, we want everything to be as neat and uniform as possible. So, but if at the end really it doesn't meet the requirements, and we cannot get the pills to dissolve within the time limit that is specified, that means potential recall for the our clients, our customer. So the customer, our customer are the pharmaceutical companies, and their customers are the pharmacy. So what happens is if we demonstrate what we call a non-compliance, that means that the company needs to call all their clients. They need to send a warning to Health Canada. Health Canada needs to publicize that and say there's been a problem with this med, this batch. It needs to be removed from the shelves, so you won't be able to buy that anymore. And if you have some of that at home, you need to bring it back to the pharmacy to get a refund.
Yeah.
And so there's a lot going on there. So obviously, it's not something that you want to happen, because the consequences are very important for the health first of the patient, and the financially, of course.
I was going to say, like, from a child's perspective, like, you know how sometimes you might get really stressed about a test upcoming? The thing that makes adults and companies and governments freak out is when they have to recall something. That's like everybody's worst nightmare as an adult. So we try at this stage to make sure that we don't have to do that, that everything is safe, everything is good. But also, really glad we live in a country when we do notice that things are wrong, that we do recall them. That's go Canada.
Exactly. And everything, every test that we do here is double checked by a second person, what we call them verifier, data reviewers, that they make sure that everything was done accurately, that all the steps were done correctly, that the method of testing was followed, that the calculations are right. Everything is double checked to make sure there was no mistake done, because the consequences are very important.
Before we wrap up and we go on to our next speaker, I just want to say a quick query on the AI front. So we've had this as a question from a class. Can AI, can modern technology help the work that you're doing in any way? Do you envision that in the next few years?
No, I would say not directly in the lab for now. There's a lot of AI more in the research part, where you can use it to help analyze a ton amount of data that you generate when you do your research. The AR on the testing part is more, it's used, but more in the background. There's a lot of documentation that needs to be done. There's a lot of procedures. So the AI helps us there to make it more efficient and standardized. But you cannot, everything that we do in the lab, so you saw all these instruments, the chemicals that we use, the solution, everything needs to be done by hand. There's some that we can automate with instrument, but there's, the curve of investment in the industry for automizing these tests is very slow, because it takes a lot of time, a lot of programmation, and a lot of money. So in the end, if a human can do it right away before, and you don't need to wait 3 years of development for the robot to be able to do it, eventually the robot may be able to treat 100 samples at once. So that will be the advantage. So this is where the industry usually aims, where they have a lot of volume, a lot of complicated steps. This is where they will introduce automation. And yes, there's a lot of talk of AI in the industry. Right now, it's not used directly in the lab, but for sure it's going to be coming sooner than we think.
Thank you very much for the nuanced answer and for the deep dive into all the cool work you do. We really appreciate having you in today. And thank you.
You're welcome,
guys. Awesome.
Thank you.
Well, we are going, as I said, we've got a train of incredible speakers today. We've got a Kahoot, we've got question periods, we've got everything. So I'm really excited to keep the train going. A big thank you to Jean-Francois and Christina for showing us the incredible precision and care that goes into every step of getting a medicine approved before it goes to our pharmacies. AI is rapidly reshaping how we discover, design, test, and deliver medications, but for AI to work, it needs a lot of data. And there's so much data right here at NeoPharm. Each of these machines is collecting data on how medicines work. It can be shared with other researchers and other labs around the globe, and it can also feed AI-powered tools. So to help us understand what this future might look like, we have Jesse Islam, a machine learning researcher at McGill University in lovely Montreal, who's at the forefront of AI-driven pharmaceutical information. So a big welcome welcome to my fellow Jesse out there. Can you tell us a little bit about the models you're developing to bring safer and more effective medications to patients faster than ever before? Do tell.
Thanks, Jesse, for the introduction. So maybe I'll start a bit with the problem that we're trying to deal with, and then how I go about solving that. For general drug development or any kind of medicine development, there's a set process in place where you first define what you're trying to target. Then you go on to do some in vitro testing. So they do like in test tubes, they're testing a bunch of things. Then they move on to mouse or animal studies where they try to understand if the medicine you created is safe. And then they move on to human studies where they try to make sure that it's also safe in humans before giving it across the world. This process takes a long time. You can think around 10 to 15 years just to get one medicine across that whole set of testing. But if we were to do this test, it turns out, interestingly, that 1 out of 10 medicine that we try to develop actually result in a medicine we can use at the end. So this means we have 9 out of 10 not working so well. And that's 10 to 15 years of work essentially thrown to the side. A bunch of animal testing, human testing even, that's maybe avoided if we can discover better targets. So the question I then have is there a way to validate or make sure our targets are better targets from the start? So for that, to make sure that a target is useful, there's common ways that we do this, but then my own work was focused on this idea of trying to make a intermediate step. So I have a set of things I want to target with my medicine, and then I have a bunch of tests that I have to do. Can I do something in between virtually? What I call virtual experiments. So this is what my research is focused on. Yeah. So, or do you want to say something?
No, I'm just keep going. I'm just fascinated.
Okay.
Sorry.
Yeah, my bad. So yeah, this is what my work is essentially centered on. This idea of creating these virtual experiments so that we can do this. So right now, you may not want to test on 1 million mice and humans,
right?
That for every single drug, that isn't ideal. But virtually, given that we have enough data, and over the years, hundreds of years at this point, where we have collected lots of wide-scale data sets, we're actually able to perform these experiments virtually and try to see which one actually performs ideally before we actually start to do the real tests, which are still relevant. We can't avoid those.
How long does it take to make up one of these virtual? Like, I mean, as someone who this is like totally, it might as well be, cuneiform to me. How, like, is there a time length for this? Can you do this in like an hour, a day? Is this a week of planning to come up with a virtual experiment? What's the deal?
Oh, there's a few steps. One, decide what data set you want to look at. So that can take anywhere from like a day to a week, or maybe you have to make it. That could take a much longer time period, but there's already a ton of data sets you can use. So once that's decided, the process itself to go through 100, for example, let's say 100, about a day-ish. Like you press a button on your computer and then you go get some coffee and then maybe play outside and then come back and then 100 experiments happen and you get to test them.
Super cool. And you mentioned, so instead of testing on people or animals, this is something that we've brought up a lot in health programs for kids. So one of the goals here is to prevent us having to use animals for experiments, or to use a lot fewer of them in experiments. Can you speak a little to that?
Interesting. So it's a downstream effect. So it's the main goal is really to make it so that the drug development process, right? 1 out of 10 drugs or medicines that we try to create are going to actually enter market, but that means 9 out of 10 will not, and there's a ton of resources dedicated to that. Some of those resources involve human testing and animal testing. So if we can get that 1 out of 10 to 2 out of 10, 3 out of 10, that multiplicative effect from getting better targets should make it so that we do less testing overall. But will we scale up? Of course, there's if you can get 10 out of 10, are you actually going to do fewer experiments?
Yeah.
It's a more complicated and nuanced political issue, I think. But yeah.
Well, we'll ask one final question before we head into our Kahoot and our Q&A, because I know we've got a lot of kids excited to start with that. But is there, like, this must be such an exciting time to be in this field because there's so much investment, there's so much interest. Just, do you get the chance to work with a lot of people from lots of different places? What does your day by day actually look like, Jesse? I'm so curious.
Oh, tons of presentations. I think a lot of me looking at a computer screen. So tons of math, So I don't know if anybody likes math in these groups, but yeah, if they love math, then there's tons of place for you and machine learning and AI. So yeah, it's a lot of computer programming, talking to people.
We are so happy to be part of your regular day of talking to people on a computer. But thank you, of all the people you could have been chatting with, that you joined us for this. We really, really appreciate it, Jesse. Awesome. Well, fine people. I know there's so much to share. We really appreciate all our speakers today. I know our NeoFarm Labs team have had to go. We are going to dive in with our Kahoot in a minute. So for those who joined in earlier, our game pin is 548-540. For those who are new to Kahoot, the faster you answer, the more points you get. And what you win is everyone here's everlasting respect. So no pressure at all. We're going to dive in with this in a second. We're going to take as many questions as we can. If you do have to go, I know some of our classes do, please do email us questions. We'll get them to all our speakers after the fact. But I'm going to get us underway with our Kahoot, and let's dive in together. Here we go. And get underway. All right. Drum roll, please. Question 1. How many healthcare facilities are there in Canada? We talked about this near the beginning. Is it 70, 700, 7,000, or 70,000? Dun, 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 dun. I think there are 70 in my town, and I'm in a small town, so that might give you a hint. It's quite a few. What do we think? I grew up in Toronto. I think we had like 3 big hospitals within about 5 kilometers of my house. So, a lot of you thought 70,000. So there are a lot, but not quite that many. 7,000 healthcare facilities in Canada. For the 15 of you who really paid attention way back when, well done. Nifty Gecko has our lead as we go to question 2. How many images do you think it took to train Scopia's AI surgery model? 100, 1,000, 10,000, or over 10,000? How many images like this did we give the model so it could make better decisions? Very cool. And I can highlight some of those later as well. 48 answers. A lot of you in the Kahoot today. Very exciting. All right. Let's see. One more question. Yes, over 10,000. So this is the thing with data, is that we've been, as Jesse said, been collecting this for years and years and years, over 100 years of images like this in the work that we do. So we are going to give it as much as humanly possible so that it can be as good as humanly possible, or as good as robotically possible, as it were. Zippy Unicorn comes in first in that one. All right. What is the main job of a pharmaceutical lab? Make the medicines taste like candy, check that medicines are safe and work properly, give free medicine to everybody, paint medicine bottles. You know, I must say, I do like medicine that tastes like candy. I liked it a lot as a kid. Now, my medicine doesn't taste very good anymore. However, I have something called the placebo effect, works really well on me. So all medicine just instantly makes me feel better because my brain tricks myself into thinking that it does. So almost all of you got this right, of course. Check that medicines are safe and work properly. There's a huge, like in Canada and the United States, for our part of our audience, or around the world, we really do as a society want to make sure that medicines heal people and don't hurt people. It's very, very important. So I'm glad we've gotten the chance to cover it today. All right. One advantage of virtual experimentation is that scientists can discover new medicines more quickly, play video games while waiting for the results, dress up the lab mice as superheroes, sounds like fun, or take more vacations in Florida. Maybe a bit of that too. But again, we're making this fairly easy to wrap up. We want to reinforce the main concepts today. So really appreciate this. Yes, discover new medicines more quickly. All right, let's see what that does to our leaderboard as we dive in. Missy Lou's class, we're coming to you first for questions. You're right at the camera. I love it. Classy Gliders is third. Fearless Rooster second. And first for all the marbles, drum roll, please, is Zippy Unicorn. We are nearing the end of the broadcast. I'm going to bring up some cool stuff highlighting the future of AI and more as we wrap up together. It's just amazing to me and reassuring to think of all the people and tests that machines that are involved in making sure the medicine that I take when I'm sick is safe and effective. And as you all expressed, it's so exciting to imagine a world where AI-powered robots can make surgery safer and even make custom medications just for me. We're working on that all over the world. AI is already changing how we go from being sick to getting healthy, and it's such a powerful tool, but it also consumes a lot of energy. And so Digital Moment has a challenge for you. Can you think of a way to design and train AI models that are both powerful and sustainable? Well, I encourage you, check out digitalmoment.org, check out the amazing 2030 Digital 2030 challenge that they have to help solve this and understand a little bit more of it. We've covered this in all our Digital Moment programs. It is so, so cool, and we'll share that with all our classes as well. And finally, we'd like to thank everyone who came together today to make this event such a success. Gabriel Reves from Scopia, Jean-Francois Delage and Christina Mihova from Neofarm, Jesse Islam at McGill University, Elsevier and the Complete Anatomy team for providing those amazing 3D and anatomy visuals, our friends at E2 Adventures for producing the event, and our friends at Digital Moment for powering this event. Thank you so much for tuning in. January 27 is our last event in the series, The Invisible Assistant. And with that, we're going to bring in all our class to say a big thank you and farewell. Second Street Mystery, thank you so much, everybody. Have a wonderful day. Bye for now, guys. Bye.